Hi, everyone. Welcome to OsteoBytes. Thank you for joining us this Thursday. My name is Christine Iptoma, and I am mom to Osteo Angel Dylan and Director of Scientific Programs at MIB Agents. And today on OsteoBytes, we are talking with Dr. Václav Ranc about treatment of osteosarcoma using smart, targeted drug delivery nanoplatforms. Thank you so much, Dr. Ranc, for joining us on OsteoBytes today. We're thrilled to have you. And a little bit more about our guest today, and I'm trying to pronounce his name correctly. I'm sure I'm not doing a great job, but Dr. Václav Ranc received his Ph.D. degree in analytical chemistry in 2009 from the Department of Analytical Chemistry at Palacky University in Olomouc, Czech Republic, under the supervision of Professor Karel Lemr. He was then employed at the Department of Analytical Chemistry at Palacký University Olomouc as an assistant professor from 2008 to 2010. And in 2011, he joined the team of Professor Rainer at the Department of Medicine at the University of Fribourg in Switzerland as a research scientist. He then worked there from 2011 to 2012, and he's been employed at the Regional Center of Advanced Technologies and Materials at Palacký University Olomouc since 2012. He has authored or co-authored 74 scientific publications, and he's a co-author of 3 international patents. His research activities are directed towards the development of nano-sensors based on the utilization of surface-enhanced Raman spectroscopy and electrochemistry, and synthesis of functionalized nanomaterials for drug delivery systems. Welcome, Dr. Ranc, and welcome everyone joining us today. If you have any questions, please feel free to add them to the Q&A, and we'll make sure to ask Dr. Ranc. And before we get started, we have some announcements and reminders. For bereaved parents of osteosarcoma angels, we do have a Healing Hearts for Parents coming up next week on August 16th at 7 Eastern. And then starting in September, there'll be a new series of Healing Hearts for bereaved parents on Wednesdays and Sundays running through the end of the year. And in addition to the sessions for the bereaved parents, we are also offering different sessions for young adult siblings and teen siblings. And I'll put some more info on the chat on how to sign up for those. And we also have our next virtual tumor review board for osteosarcoma, which we affectionately call Turbo, on September 13th. And that's a Wednesday, 5 p.m. Eastern. And it's a really great educational forum. It's open to clinicians and researchers interested in osteosarcoma. And if you're interested in attending Turbo or presenting a case, I'll put a link in the short form to enter your information into the chat. And I should also say that if you're a caregiver or a patient, while you can't participate directly in Turbo, if you're interested in having your oncologist present your case, you can certainly share that information with them. And then we are gearing up for Childhood Cancer Awareness Month in September. So Dr. Ranc, every September, I guess actually this might be a global thing, Childhood Cancer Awareness Month. But with a $25 donation, you'll receive a beautiful big gold bow that you can adorn your mailbox or front door with to raise awareness for pediatric cancer and show support for families impacted by pediatric cancer. And all the funds raised from the bows will support programs, education, and research for the osteosarcoma community. And we would like to thank the sponsor of this episode, BTG Specialty Pharmaceuticals. BTG provides rescue medicines typically used in emergency rooms and intensive care units to treat patients for whom there are limited treatment options. They are dedicated to delivering quality medicines that make a real difference to patients and their families through the development, manufacture, and commercialization of pharmaceutical products. Their current portfolio of antidotes counteracts certain snake venoms and the toxicity associated with some heart and cancer medications. And their drug, Varaxase, is for high-dose methotrexate toxicity, which, of course, high-dose methotrexate is kind of one of the standard drugs used for osteosarcoma. Thank you, BTG Specialty Pharmaceuticals and Dr. Ranc. I am done with my announcements, so I'm going to hand it over to you to start sharing your presentation, and we'll get started.
So first of all, I would like to thank you for inviting me here to this event. I would like to introduce to you our consortium based on the Nanoplatforms project. So I will talk about this for a few minutes, and then I will go to results what we obtained in this project, which is related to treatment of osteosarcoma. So first of all, Nanoplatforms actually, as you may be able to see on our website, is acronym for the Advanced Hybrid Diagnostic Nanoplatforms for an Active Drug Delivery in Cancer Treatment. So that's the name of the project. That which actually led to the formation of our consortium. But first, before I begin with what we do, actually, I have a few slides about Osteosarcoma. You probably know better than me what this actually means, but just a few hints or few important points. So, Osteosarcoma is a malignant spindle cell sarcoma, in which the malignant cells produce osteoid or bone in the background of sarconomastoma. So, the important point is that this mainly affects the children or teenagers, so that's why it's important for us. That's why we are focusing here, because there are, of course, other types of cancer, but where different teams are doing their research. But for me and for our team, osteosarcoma is the most important one, because it affects the children and teenagers of the young generation, which is very important to us. So the treatment is usually not easy. That didn't change much until 1970s. The treatment was done based on the surgery, and it wasn't very good, and the efficiency also wasn't great. But since then, many things changed. So we have now some modern treatment approaches, including chemotherapy, immunotherapy, bisphosphonates, radiation, or still surgery therapy is applied in some cases. So I highlighted two drugs here. I highlighted doxorubicin and cisplatin. Why? Because those two compounds are the ones who are of our interest, and we will use them during our project, and I will show the results we obtained using those two compounds with our nanoplatforms. But before I get there, I will start with what actually Nanophoton is, and what we are doing, and why we are here. So, Nanophoton project is composed of different people, different scientists working in the chemistry, biology. We have medical doctors who all share their extensive expertise to find one mutual aim. And that's to deal with the fundamental issues of nanotechnology-driven anti-cancer drug delivery. So why we are doing that? Because the chemotherapy or the drugs which are served now or which are used are toxic. They are toxic to the osteosarcoma, that's true, but they are toxic also to our bodies. So that's not what we want, actually. We don't want to inject into people something which is toxic or doesn't work. So what we want to do is to target this delivery, to transfer this drug from the point of the injection directly to the point of care, so to the osteosarcoma, to the cancer cell lines, and treat it there. So it's not affecting anything else in the body. So that's why we use these nanoplatforms, which I will show you later, to bind the drugs and to actually magnetically or using any other force move them directly to the cancer cell lines in the body, and it's affected there. So this is the main aim of the project. So the consortium, the team is composed actually of 3 sub-teams. Teams presented in Palacký University. That's me, and this is our team. And we are a team which is the expertise of our team is the synthesis of the nanoplatform. So this is what we do. We are synthesizing different nanomaterials, different nanoplatforms. But that's only part of this problem. We need the drugs. We need the drugs to put it on the nanoplatform and to have it working somehow. So that's why Manu University comes into the project. In Manu University, there is a team led by Dr. Montañer, who is an expert on the synthesis of anti-cancer drugs based on cisplatin. Cisplatin is a complex usually used for the chemotherapy, but what they do is kind of novel, because they have a procedure how to functionalize the drug using different functional groups, so we can attach them on the nanoplatform. So this is not the cisplatin which is currently used in chemotherapy, but this is let's say it has the same effect, but the different functionalization, so we are able to attach it on the nanoplatform. So when we have the nanoplatform and the drug, there is the last step that needs to be done, and that is the testing on the cellular lines. So and this is where CNL, or this is the research institute in Italy, comes in. So this team is led by Dr. Panzeri. I will show you later. And they are experts on the study of interactions of nanomaterials with cells. So we have the whole package. We have the team which is expertise in the synthesis of drugs. We have a team which can use these drugs and put them on the nanoplatforms. And then finally, we have a team who is able to evaluate the function of the nanoplatform on the cellular lines, both in 2D and 3D models. So we are working on 2D cellular lines, which is in the Petri dishes, and also 3D models, which are very close to actual tumor in the body. So we have the information also. This system works also or it could work also in the real scenario, and not only in the Petri dish. So, but now I would like to make a short detour and tell you something about the Czech Republic, because it's just a few words, but maybe you don't know where this, or you know only a little about it, so I'd like to show where it is. We were part of the Czechoslovakia. This is maybe close to, or maybe you heard it somewhere. But this was until 1992. Then there was a divorce. So since January 1993, we are only Czech Republic and Slovak Republic. We are part of the Europe actually. So as you can see in the map, and we have a robust democratic tradition, highly developed country. Actually, that's a nice word, but practically, the let's say economy can be very well compared to Italy or Spain. So we are on the same level here. Czech Republic is not very large. There's only 78,000 square kilometers, and the population is 8.5 million. So this is not even one of the biggest cities in the world. So that's a high population, and actually 10% of people living in Czech Republic live in a Prague, which is the capital city of Czech Republic. And also we can say that's one of the most beautiful cities or towns in Europe, because and that's actually nice, because we have all the monuments focused on very small area. So when you go there, you have all the things around you, and you can just walk there. You don't have to use a tram or train or anything. It's just there. So life expectancy in Czech Republic is 74 years for men, 80 years for women, but that's just for the curiosity. That's not important. So this is about Czech Republic. Palacký University is located in Olomouc, which is on the eastern side of Czech Republic. It's actually the second oldest university in Czech Republic, with a huge tradition, or large tradition. And this year we are celebrating 450-year anniversary of our university. So it's quite old, but it's also modern. We have more than 20,000 students, which you can imagine that you can, that's comparable to the seating capacity of Madison Square Garden. So you can move all the students there, and they can all sit there. We have 840 accredited study programs. So that's a lot, and those are present in 8 faculties. So besides the Faculty of Science, we have medicine, and we have history, and we have other faculties. So this is about the Palacký University in Olomouc, and you can see that this picture is for the rectorate. So this is where the rector's office is located, in the second floor of this building.
But this is like a little recruiting. If there are any positions open in your lab, you can live in beautiful Prague.
Yeah. Actually, we have some positions, so I can forward you the advertisement if we'd like to. So But I'm coming to the lab in the next slide, so you can see what we are actually doing there. So Palacký University has a Faculty of Science, and the Faculty of Science is a regional center of advanced technologies and materials. This sounds very fancy, but what we do here is that we are synthesizing, developing various nanomaterials for applications starting from energy storage for batteries and to biological samples or to biology, where we are using the nanomaterials to analyze biological samples and doing that delivery system. So the whole portfolio is there. So we have one group which is focused on the environmental application. So they are using nanomaterials to treat the environment, to clean the water and to clean the air. So the focus is really broad. We have over 100 scientific team members with a level of internalization exceeding 35%, which means that the main language in our institute is English. So that's the only way how you can actually communicate. And which is actually quite new for Czech Republic, which was until recently closed. So and the island in the middle of Europe. But now we are opening, so which is actually great. So we are trying to we have people from all countries of Europe probably. So that's great. So for the materials we are synthesizing or we are preparing, they are usually based on carbon. So we have carbon-based nanomaterials, including graphene, which you maybe heard something about that material, but still I will show you later. And its derivatives like graphene oxide, reduced graphene oxide, and graphene acid and graphene amide and so on. Also, we are producing quantum dots. So those are actually chemicals which are used nowadays in TVs as a lighting. So instead of the LEDs, you can have quantum dots which emit white light, and that actually the TV is brighter than using the LEDs. So you can buy that from LG probably or some other companies. And we are also synthesizing magnetic nanoparticles. So this is used also for their delivery systems, but for different projects. But also for MRI applications. So as a contrast agent, if you go to MRI, if you have some problem, medical issue which needs MRI to diagnose this problem, you can use magnetic nanoparticles instead of the classical compounds used now. So we have a better resolution of the system. So the infrastructure of the center consists of let's say, I said, well, it's fully equipped, but let's say the equipment is very good here. It's on the top level regarding the microscopy, because we have several electron microscopes, including high resolution transmission electron microscope, which was the first one in Europe actually when we bought it. And we have some other techniques, including AFM. That's actually the technique I will talk about later. So that means atomic force microscopy. This is used for the evaluation of the high profile nanomaterials. So if I'm talking about graphene and I synthesize it, I always need to know what's its high. If it's 1 nanometer, 5 nanometers, or how thick it is. So AFM serves to tell me this information. Because from electron microscopy, I can see what's its size. So if it's this big or this big, but I don't know how thick it is. So for the thickness, we have atomic force microscopy. Squid and BET, those are techniques used for the magnetic characterization, or if to know if the material is magnetic or not. And BET is for the adsorption. So we need to know what's the surface of the material, because for the graphene, batteries, for instance, we need this surface to be as high as possible. So we are able to produce material based on graphene where 1 gram has a surface of a stadium. And that's true, actually. So, and BET techniques helps to tell us what's the surface of the material, actually. Raman and infrared spectroscopy, those are used for chemical characterization of nanomaterials. So it doesn't have to be nanomaterial, but for different kind of materials, we see what's the chemical composition based on the data from Raman and infrared spectroscopy, and I will get to that also later. So, and in this building, we have a group which is called Nanomaterials in Biomedicine. This is my group, actually. And the primary goal is to design efficient nanoplatforms and establish new diagnostic protocols to address several diseases. So, to say it in other words, we are developing nanomaterials for the diagnosis of different diseases based on the detection of markers. So we are able to develop new analytical systems to detect, for instance, dopamine, serotonin, and some other compounds. So we can use that as an information for some psychiatric diseases. And we are also developing 2D nanoplatforms as a drug delivery system. So those are two different roles of the group. The detection systems are usually based on the optical methods and plasmonic nanostructures. The plasmonic nanostructure, maybe that's a new word, so I will try to explain it a little bit. Surface plasmon is a property of nanomaterial which is actually known for hundreds of years. But nobody knew that this is the one that didn't exist. But if you see the window in the church and it's red because of the gold in it. And if you imagine a gold brick, it's yellow, it's shiny. And if you destroy and keep destroying this brick until the smaller and smaller pieces, and you will end up with nanoparticles, which are like 20 nanometers, or let's say in nanometers scale, the color changes. You can have red, you can have blue, you can have violet, you can have whatever you want, actually, and it's still gold. The only difference between those red gold particles and the brick is the size. So if you go small enough, you will end up with red particles which are actually used to color the glass in church for hundreds of years, but nobody knew it's nanoparticles. But that's the plasmon. So we are using this optical property of this nanostructure to detect analytical or to detect some compounds in clinical samples, including dopamine, serotonin, immunoglobulins, and some other compounds. So on the right, on the figure, you can actually see one of our nanomaterials when we have silver nanoparticles, which are around 30 nanometers. So silver also shiny metal. But if you go low enough, or small enough, with the material, you can see that the color changes to red or to yellow. And this is the image obtained from the electron microscope, and this is sensitive to elements. So the silver nanoparticles are red, but not red because they are red, but because this instrument said that they will be discolored. So we can see how those nanoparticles are organized on the surface of the NFE, that stands for the magnetic nanoparticles. So this is the system composed of magnetic nanoparticles here in blue and silver nanoparticles, which is used as a detection system for dopamine in this case. So this is one of our products, actually. And we are using that. Yeah.
Oh, I just ask you. So there's the three diseases that you know that you're studying here: osteo and adenosarcoma and glioblastoma. Is there? And I know you mentioned for osteo it was because it affects AYA pediatric patients, but is there anything about those three diseases that kind of make them good diseases for nanomaterials and nanoplatforms?
Yeah, actually, yes. I will get to that in the results, actually, in 2 slides.
Okay.
Just an introduction into before I get to the osteosarcoma, but I will get there. But of course, we are developing nanomaterials to treat osteosarcoma and also glioblastoma and breast cancer. So I will show you the results in few minutes. Okay. Yeah, I know the time is running fast, so I will maybe speed up. So that's it for the Czech team. now a few words about the team in Italy and Ireland. So in Italy, the part is located in ISTEC, which is the Institute of Technology and Science and Technology for Ceramics. So this team is led by Silvia Panzeri, and she's expert on the study of interactions with nanomaterials and the platforms with cell lines. So including Osteosarcoma and glioblastoma, they have all these cell lines present in their lab, so they can test our nanomaterials. And in Ireland, there is Diego Montañer, who actually has two other team members, Daryl and Maria, who are synthesizing the ducts, and they are sending it to us to Olomouc. We usually pick this duct and immobilize in the platform and send it to Italy for testing. So this is the basic workflow of our work. So for the objectives, the project is mainly focused on the early stage researchers. So this means PhD students and master students who can travel to Italy or to Ireland to get the work there and to work on their thesis. So that's how we are actually sharing the expertise or the knowledge. But the main goal of the project is, of course, to develop a product, to develop something which can be used as a drug delivery system to treat the patients. Right now, we are doing the research. We are doing in open way, so that means that all the results that we produced are open and available to everybody. And also, that one good thing about the project is that we are learning from the, let's say, from the more advanced partners in Italy and Ireland how to manage the project, how to manage the consortium, and so on. So this is the last slide actually of the introduction, and now I will get to results, what we got and how we got there. So this is the basic workflow. So to treat the osteosarcoma, we need the drug delivery system. Because we don't want to put the platinum complex directly into the patient, we want to deliver it through the body to the place where it needs to be. So we are using graphene oxide, which you can imagine as a very, very thin material, only one atomic layer high. So that means that this is also razor sharp, and that's not safe. So if you put this into the petri dish with cell lines, they will die because they will be cut. So that's why we are functionalizing with polyethylene glycol, which is a compound which is softening the edges of the nanomaterial, and this makes it much more biocompatible, and actually the cells are they're okay with that. So it's not toxic at all. The polyethylene glycol is not one compound, it's actually a family of compounds. We are using the 8-arm polyethylene glycol that is highly branched, and why 8-arm? Because on every single arm of this material we can attach the platinum, so the loading capacity is much higher. We can load much more platinum on the material thanks to the more bonding places present on the arms of the polyethylene glycol. So this is the basic workflow, how this is synthesized. So first step, we have the graphene oxide, then we have graphene oxide with polyethylene glycol on the surface, and the surface is then functionalized with the platinum complex. And those nanoplatforms are then tested in Italy in cell lines for breast cancer, glioblastoma, and osteosarcoma, just to see how it works. Those are the images from the atomic force microscopy. Like I said, this is the technique used to see the high profile, to see the high of the nanomaterial. So you can see on the image A, the graphene oxide we developed or we synthesized, and on the image B you can see the high profile, which is somewhere around 1 nanometer. So the high of those flakes present on the figure is around 1 nanometer. We also used the Raman spectroscopy. This is the image on the left with D and G labels, and this tells us what's the structure of the material. So those D and G bands, actually the ratio changes with the changed chemistry on the surface. The more structural groups, the more groups which can be functionalized, usually carboxylic groups, OH groups, and some other groups, which are on the graphene oxide. So their content actually changes with the D band. So the higher the D means that there is a higher number of structural defects caused by these functional groups, which is great for us because we are using those groups to attach the polyethylene glycol. So this is just the for us. This is just the let's say information that this material can be functionalized with polyethylene glycol because it has enough functional groups on the surface, and the height is around 1 nanometer. But for the size, we are using also AFM, but in different way. You can imagine that we measure the sample of the 2D material on 12 different places. Each place is 50 by 50 micrometers, and we statistically evaluate the size of the material present on in this measured area. And you can see that most of the particles or the flakes of the graphene oxide falls in the range between 10 and 100 nanometers, which is great because if you want to deliver something into the cell, it has to be around 100 nanometers, because if it's bigger, it's much more difficult to get it into the cell. So the aim here was to prepare something which will be smaller and also much better for the cell, so they can accept it and it can work there. Then we functionalized this pristine graphene oxide with polyethylene glycol. As you can see on the right side, we are using infrared spectroscopy just to confirm that the polyethylene glycol is on the surface. This is shown on the figure STIR. So the STIR, it stands for the Fourier transform infrared spectroscopy, which is a technique which is saying us what's on the surface of the material. So if we measure the pure polyethylene glycol, you can see we have some signal, and we measure the graphene oxide together with the polyethylene glycol. We see that the polyethylene glycol is present on the surface. So this is a confirmation. The second confirmation is that the high profile of the material changed. It's now thicker, and it's thicker because of the presence of the polyethylene glycol on its surface. So this is how we prepared the nanomaterial. And now regarding the cell viability and morphological analysis. So if we use this nanomaterial together with the drugs on its surface, this is for the osteosarcoma cells, after 72 hours of the interaction, you can see on the left side that we have three different cell lines: U2OS, MG63, and SAOS2. Those are three different cellular lines representing osteosarcoma. And you can see if we use just the nanomaterial with the polyethylene glycol, which is here labeled is GO at PEG, it's not toxic because there is no effect on the cells. It's around 100% of the cell is still somehow without impact. But if we add the platinum only, part of the cells will die, which is actually okay. This is what we want to kill the cell lines. But if we add the nanomaterial together with the platinum with the same concentration, the effect is much higher. So compared to the pure platinum, so this shows that there is some synergy between the drug delivery system and the platinum on its surface, because the effect of this drug delivery system is better than compared to the platinum itself only. It's also shown on the figures from the Fluorescence microscopy. So you can see the cells there, especially you can see it on the GO at PEG-PT, that stands for the polyethylene glycol together with platinum. And you can see that the number of cells is much lower. So this means that after 72 hours, not all, but many of the cells are dead already, compared to the platinum-free or the nanomaterial itself, because there it's you can see that there are many cells and they are overlapping and they are almost intact. But when we add the nanomaterial, most of the cells is gone. So that's for us good information that it works, because this is what we want. Now the second part will be to target or to transfer this drug to the osteosarcoma actually in the 3D model.
So can I just clarify? So the PT free, so that's basically just cisplatin on its own compared to the GOAP head, which is platinum loaded onto this pegylated graphene oxide.
Yes. Yes, and the similar results are obtained for the glioblastoma, even though it's not so prominent, because even platinum-free complex works here very well. So the results are comparable between platinum-free, which means that we edit just the complex without the platform. But good news is that the platform works as well. So if we see the cells only, so this is untreated, this is the control. The cells are there without any effect, and we at the graphene oxide functionalized only with polyethylene glycol. You can see that the cells are not affected at all also. So the material itself is non-toxic, but if we edit platinum or platinum complex, it's highly toxic to glioblastoma cells. And similar results we obtained for the adenocarcinoma cells, so for the breast cancer. So here you can also see that the graphene oxide together with the polyethylene glycol and platinum works better than the platinum itself. And you can see it on the figure of the histogram, and also in the results from the fluorescent microscopy. So those are 3 tests we actually did, and we see that our nanomaterial works even at lower concentrations of 15 or 30 micromolars. So this is good news. So the next step actually was to add one more drug on the surface, because this is a 2D material, so we are not limited only to one drug. We can do multi-drug system. So we added also doxorubicin as one of the potentially very good compounds to treat osteosarcoma. And you can see on the figure, which is labeled MG63, maybe you remember that this cell line was also present in the previous model. And here we are comparing platinum-free for the controls, doxorubicin together with platinum, and cells only. Cells only, this is negative control, and then we have controls only with the drugs without the platform. And for the platform, we have graphene oxide with polyethylene glycol, with platinum and doxorubicin. I don't know why it's twice there, but it's actually same. So with platinum and platinum doxorubicin. And if the platinum and doxorubicin mount on the bind on the platform is labeled here in the graph with green color. And you can see that in all the cases, the most of the cells is dead for the green color. That means that our buffer is the most efficient, even more efficient than if we add the doxorubicin and platinum themselves without the platform. So for the 15 micromolar, our platform works, and you can say that the doxorubicin free, this is the violet one, and platinum, the doxorubicin red one, and platinum free bright blue, they don't work at all, or almost. There's almost no effect for the platinum. But when we bind this platinum on the surface of the nano platform together with doxorubicin, the effect is there. So this also works very well. and also for the concentrations from 15 to 60 micromoles. And again, for the nanomaterial itself, which is here in the brown color or red brown, there is almost no effect. So this means that still this nanomaterial is perfectly safe for use because it has no effect on the cell lines.
So am I reading this correctly if I'm looking at the higher concentration, like at the 60, that there isn't as much of a difference between the GoPEG cisplatin doxo combo versus cisplatin doxo free, the GoPEG doxo, and why is that, do you think?
I believe that the concentration is simply too high, so it will just it will work the same because I think there is an effect that it will the sum of the cells will survive no matter what. So for the green for the platinum complex, so it works on the some level, and it seems that there is not much difference between the concentrations because it works almost the same or statistically the same for all three concentrations. But what changed is the effect of the platinum free and doxorubicin free for different concentrations.
Okay, thank you.
I'm not sure about the mechanism. Actually, that's the question we ask ourselves, but that's what we are doing now, trying to go behind this graph and see what's the mechanism of the action. So maybe I will have a better answer in the future, but right now that's the only one I got. So
okay, thank you.
Well, thank you. Okay, and now for the 3D models, actually, you can imagine the 3D model as a hydroxyapatite nanocrystal on a collagen scaffold, which looks like the image in the center or lower center. So this is the 3D model of the tissue that we are using this to load the cells into this and treat them. So you can see that we have different. This was done on the osteosarcoma cells, and we loaded 30,000 cells on this 3D model, and saw what will happen in time. So we measured the penetration of the cells which were actually injected on the surface after 3 days and after 7 days. After 3 days, you can see that the cells grow into the lower levels of the model, and 7 days it's even more obvious that the cells are like everywhere. So this is or not everywhere, but they after the 10 days that they will be. So so we are using this model actually to treat or to test the function of our systems. But right now we can only see the DAPI. It's for to see if the cell is living or not, just to evaluate the function of the 3D model. But this is where we are now. So right now, actually, or not now, but this month we will test also these platforms on this model. So we will have the results from that. But for now, we are here just to see if we are able to use this new model for osteosarcoma, which we probably are. So that's actually all for the results. So at the end of the presentation, I have few articles that few scientific papers we published during the run of the project. So you can see, and maybe if you are more interested into what we are doing, so you can read those articles. The first one is on the products for treatment of osteosarcoma. This is for the platinum complexes themselves without the nanoplatform. The second paper is like I said before, that we are also developing some analytical systems for the detection of various markers related to cancer. So this is for the prostate cancer. This is not related to osteosarcoma, but to prostate cancer. but we developed a system which is able to detect PSA and free PSA in one analysis within minutes, so we can say what's the status of the patient, if there is any need to for treatment or for further diagnosis. The third paper is actually for the materials I showed before, for the graphene oxide nanoplatforms, which enhance the effect of the cisplatinum-based complexes based on the synergy between the cisplatinum and the drug delivery systems. And the last one is for the cisplatinum complexes functionalized, or they have also the sugar moieties on their surface. So if we add sugar to these cisplatinum complexes, their penetration into cells is higher, so the efficiency is also higher, and this is somehow covered in the paper. And finally, I would like to thank you for the attention, and thank you for inviting me here. And this is actually our work group from Italy, from Ireland, and from Czechia. And this was actually taken last year in October here in Olomouc, so you can see that we have also nice weather and nice parks in the campus. So thank you once again, and if you have any questions, just ask.
Great. Thank you so much, Dr. Ranc. We actually did have a question come in about, have you tried using this in vivo?
Not yet. That's the plan. Actually, now we are trying to use it in 3D models, and when we have good results in 3D models, we will use it in vivo.
I was reading, and I think this might have been in kind of one of the papers, or kind of a paper where I actually kind of first learned about your work. But I think in that you had talked about that there were some limitations in using animal data to predict human response. Just because of how the nano platform worked in other species. So I was curious about that, like about being able to test this in mouse models or even canine models.
Yeah, we will use it. But because this is easy to do. You are right that it's not easy to actually use the results from the mice to human. But still, at least it's something living. So we are definitely trying, or we will use our nanosystems in mice. Actually, we have mice models here directly in Olomouc, so probably we will use that. But I don't want to go into this step before I have good results from the 3D models. So because I don't want to skip that 3D model just to have a better chance that it will work also for mice. So not to do this experiment, which is timely and costly, to do it without any solid background.
Yeah, so the plan is to, once you complete your experiments on the 3D models, then move to...
Yes. If we pass the 3D models, or if the nanomaterial, we pass this test, and we will continue on mice. Probably that's more likely.
And so I was curious about, because of the diseases that you were focusing on. Again, it was the adenosarcoma, glioblastoma, and osteosarcoma. And so, did you choose those three specifically because cisplatin is used in standard treatment, and you were looking at cisplatin specifically?
Yes, actually that's true. Because we started with the material we have, and we looked on the diseases we can target. But osteosarcoma we picked for the reasons I described before, because I believe it's important. But we see that the cisplatinum is also used for other types of cancer, so we also added those two.
And you had mentioned also that you can add multiple drugs onto this delivery platform. So, for example, you added doxorubicin. So how many drugs can you add, and would you look at then just adding, so for example, in osteosarcoma currently today, the standard is doxorubicin, cisplatin, but also high-dose methotrexate. So is that something that you would look at, of combining all three drugs?
Yes, Yes, that's actually the plan. But the third one probably will be actinomycin D. So that's one of the potentially good drugs. So that's the third candidate for the system.
And is there a limit on how many drugs you can add onto this platform?
Yes, the limit is the loading capacity, because the loading capacity of the nanomaterial is limited. So the more drugs we will add, the lower concentration we will get. So that's why we are actually right now testing what's the lowest concentration we can add to have the effect. We see that 15 micromolar concentration works in some cases. 30 is safe. So to achieve at least 30 micromoles concentration, actually this is the number which defines how many drugs we can add. But I will end up with, I think, 3, but we will have to consider also what kind of drugs, and because not all the drugs have the same affinity to the platform, so they will not bind same. So for now, the plan is to add one more, which will be the actinomycin D, and then we will see if we add more. But also that's one way where we can go. But the second one, and that's also what we are doing, but still the results are preliminary, is to add not the drug, but to add the selector. So selector which will selectively bind to osteosarcoma cells and attach there. So for now we are trying to add hyaluronic acid, which is a selector for CD44 receptors. So we have some results there, and that's probably the way where we will go. So to add more drugs and also to add the selectors.
Right. So it's more targeting, because it seems to me, I mean, when you're using drugs that are, currently being used, that the advantage is more precise delivery, which means that you could potentially have higher dose and less toxicity.
Yeah.
And so, which it seems like, I mean, do you get that effect without the selector anyway?
Yes, the data I showed you were without the selector, and you can see the efficiency of the drug bound on the surface of the nanomaterial is higher compared to the free drug. So the role of the role of the platform is there, and with the selector it's even higher. So
got it. And so given though the I mean, I know you can't really do this so much in I mean, or maybe you can in vitro. But are you also looking at and measuring any sort of just kind of the decrease in toxicities? So that if it's targeting specifically the osteosarcoma cells, that you don't see anything in like surrounding healthy tissue?
Yeah. That probably we will do with the 3D model, where we can actually add more than one cell line. So we will add also the fibroblast probably as a control, but that's also the plan. So that's not something we have now. But for the 3D model, we will also add some normal cell lines, or let's say healthy cell lines, to see how they are affected with the platform.
And then one other thing I wanted to ask about was, I think in this other paper, one of the things that was mentioned is the ability to also inhibit migration of the cells. So could you talk a little bit about that?
Yeah, that's actually an important fact for the study and for the evaluation of the work of the nanomaterial. So we did those tests. So for the migration, I think it's the third paper we published with the cisplatin and platinum, cisplatin and graphene oxide nanoparticles. So for the cell migration, you can see that there is a positive effect of the presence of the platform on that. So it's actually inhibited. So.
Right, because that would could basically help minimize metastasis.
Yeah. There's an inhibition of the mobility of the cells present where we injected also the nano platform.
And have you studied that did you see that in other diseases as well?
We tested on all three, but I think it was only in one case when we saw that inhibition.
And was that in osteosarcoma?
I think it was osteosarcoma. In the glioblastoma, it didn't work at all, actually, but and for breast cancer, the results were not very convincing, but for osteosarcoma, it worked quite well.
Because, I think, like, if I am kind of going back to the slides that you showed, the images of the cells comparing, it also seemed like it just had the biggest effect on the osteosarcoma cell line.
Yes, that's true.
Well, that's promising. Well, we look forward to results from your 3D models and hoping that moves into mouse models. And can't wait to hear more about that. Thank you so much for joining us today, Dr. Ranc, on Osteobites, and for making it better for osteosarcoma patients and really all cancer patients. More information on this and all Osteobites can be found on the MIB Agents YouTube channel, on our website at mibagents.org, and at your favorite podcast place. And next week, we're actually dropping a new episode of our AYA podcast, Osteo. So next Thursday, August 17th, keep an eye out for it. Osteo co-hosts Camille and Mia are joined by other members of our Junior Advisory Board, and they address questions that were submitted online and also some that were asked during the amazing patient panel that we had at our Factor conference recently. And so look for that, again, on your favorite podcast place, on our website, and I'll drop a link in the chat about that. And then we're going to be back with Osteobites on August 24th. We're going to be talking to Dr. Mark Mamula from Yale University. And Dr. Mamula will be discussing EGFR and HER2-based immunotherapies for osteosarcoma, hemangiosarcoma, and other cancer types in canine patients. And mechanisms of tumor growth and therapy will be discussed. You can find our Osteobites lineup for the next few months on our website. And if you have any ideas for future topics that you'd like to hear about, please share them with us at events@mibagents.org. Thank you so much, Dr. Ranc, especially for staying up at this late hour to join us live on Osteobites. Really appreciate your...
Thank you for this opportunity.
Yeah, thank you. And we hope to see everyone back here on August 24th when we talk to Dr. Mamula. Thank you, everyone. Have a good rest of the week.